We've next got to welcome Professor David Brough. So he's lead for the Neuroimmunology branch. So David did his PhD here in Manchester with our President and Vice Chancellor, Professor Dame Nancy Rothwell, and he established his own research group in 2008. So since then, David has actually won 2 UMIP awards for innovation for generating small molecule inhibitors of inflammation. David has a special interest in cytokine-driven inflammation and neurological disorders and has done some really seminal work looking at the role of inflammasomes in neurological disease. So David's going to talk to us today about the Neuroimmunology branch.
Thank you very much, Lizzie, and also thank you very much, Tracy, for allowing me the opportunity to lead neuroimmunology in this really exciting new institute. So it was, for a long time, the brain was considered an immune-privileged organ, but we know this now isn't the case. It's really important in, it innervates tissues and regulates macrophage phenotype and responses, but it's also subject to immune responses, humoral and cellular, during disease and other processes, but in disease we know that these immune and inflammatory processes contribute to a worsening of disease and our therapeutic targets. So, who are we? Well, we're basic and clinical researchers. We span basic molecular cellular biology right through to clinical trials and clinical research. And I've tried here to highlight the different aspects we do. Basic glial biology, preclinical models of brain disease and model organisms, and also pure cellular immunology. And these interact strongly with clinicians looking at acute cerebrovascular disease, neuro-oncology, psychiatry and mental illness, but also clinicians looking at autoimmune and autoinflammatory diseases, where these diseases in peripheral tissues and organs can affect the brain. And I'll give some examples of this as we go on. So I'd like to start by giving a couple of research highlights where we've shown how targeting the immune system can affect brain disease, and then give some more information on the main USP of the group. So this is research from my group, and what we were looking at is the inflammatory response, or the inflammatory contribution to Alzheimer's disease in this model. Now, Alzheimer's disease is a devastating condition. 800,000 people in the UK are affected by it, and it's a horrible disease, and there's no treatments for it. And research from our group and others have identified the inflammasome, which Gloria described in the cellular immunology, as a potential contributing factor. And you can see here on the right-hand side, we discovered that phenamate NSAIDs, these are non-steroidal anti-inflammatory drugs of the phenamate class, could be repurposed, and they target the inflammasome, and they block it. And they do this by blocking chloride channels. So we were able to establish this bit of biology that hadn't been seen before. Using the triple transgenic model of Alzheimer's disease, this animal develops cognitive problems as it progresses. We gave these animals the phenamates, and we find that when we looked at memory, wild-type animals can do the memory task. The transgenic animals can't, but after 2 weeks dosing on these phenamates, the memory is restored. So these phenamates could really target the cognitive problems in this model. And this was through an effect on inflammation. If you look at these images down here on the bottom left-hand side, these are stained brain sections where we're looking at microglia. Microglia are like the macrophages in the brain, and typically at rest they have a small cell body, elongated processes, but as they get activated, the expression of surface markers increases, and they become large cell bodies, and they start clustering around debris or pathological hallmarks, in this case such as amyloid beta plaques. And what we found was that in the particular area of the brain involved in memory, we had activated microglia, and those that had been treated with the phenamate NSAID, they were completely resting. So this showed us a couple of things. Firstly, that inflammation is a disease-modifying process in this model of Alzheimer's disease, and we think this could be really important going forward. And there's the potential for repurposing anti-inflammatory drugs used elsewhere in diseases such as Alzheimer's. Another research highlight I'd like to show, and this is related, and this is research by Kevin Cooper and from his group and his PhD student Patrick Strangward, Patrick's now an AR UK fellow in the department. Kevin's lab works in cerebral malaria, which is an important consequence of malaria, and it actually is the leading cause of death. And what Kevin had discovered this cerebral malaria is treated with anti-malarial drugs. A consequence of this anti-malarial drug treatment is some nasty side effects and a high level of mortality. And what Kevin had found, and Patrick had found, was that when they give these anti-malarial drugs, they get these inflammasomes formed in the brain, and we can study this. So these anti-malarial drugs are having an inflammatory effect. And what Kevin and Patrick discovered was that when they treated the animals with anti-malarial drugs and they co-administered an inflammasome inhibitor, or interleukin-33, which they discovered downregulates inflammasome genes, the mortality and the side effects were inhibited. So what this suggests, that anti-malarial therapy could be improved with an inflammasome inhibitor as an adjuvant, as an adjunct. So these examples show where we can target the inflammatory response, or preclinical work where we're targeting the inflammatory response in brain disease. However, one of the main core strengths of our group is the study of how multimorbidity can affect the brain. So most people who suffer a disease or a disease of the brain are usually presenting with more than one condition. So they're comorbid or multimorbid. That's a number of other conditions present together. This slide here is showing work by Catherine Lawrence. And Catherine Lawrence and Stuart Allen in the Neuro group really pioneered the use of comorbid or multimorbid animal models to study brain disease. And what Catherine's done here is she's taken the non-TG, these are wild-type animals, the triple times TGAD. This is a transgenic model of Alzheimer's disease, and Cath was studying the interaction between comorbidity and disease here. And what Catherine did was she fed the animals a high-fat diet to make them obese. So this overlaps with Tony's work previously on the cardiovascular work. And by making these animals obese, Catherine discovered that there's an effect in the brain. You can see the microglia here, small cell bodies, elongated processes in the wild-type control. They're resting. When the animals are obese, the microglia are activated. The brains are inflamed. And this has a consequence. So Catherine was looking at Alzheimer's disease, but I really just want to highlight here that this is a memory test. This obesity affects brain function. This brain inflammation affects brain function. The animals can't perform as well in this memory test. And this links really well with other research, clinical research, going on in the group. So this is work by Bill Deacon, who's a psychiatrist and a neuroimmunologist in the Neuro group. And what Bill had discovered was that in conditions of multimorbidity, obesity is really comorbid with depression. Okay? So there's inflammatory factors, there's neuroimmune signaling occurring, we believe, that's linking peripheral inflammatory disease or conditions like obesity and mental health. We can study this. We can study microglia in the brain. We use imaging such as PET imaging. These are two studies. One of them is from Manchester, showing that inflammation is present in the brains of people with depression. You can see an increase in microglial activation, people with depression or major depressive disorder. So what I wanted to highlight with that is we can study comorbid conditions in preclinical models clinically and start to dissect the neuroimmune mechanisms that underlie these processes that affect the brain. So to summarize, this is the aim of our group, is to understand complex neuroimmune signaling that affects brain functions in conditions of multimorbidity. And if we can understand these processes, we can target this. And I've given a couple of examples where that's happening. We have researchers, basic and clinical, studying brain disease conditions such as stroke, Alzheimer's disease, how the inflammation arises that drives those conditions, how it also contributes to mental health, fatigue, anxiety, depression, so on, but also how this affects peripheral disease. And here we have clinicians working on autoinflammatory, autoimmune disease, and how this affects the brain. There's also intervention studies where we're targeting inflammatory targets in clinical trials where mood, fatigue, other mental health aspects are endpoints, so we can really start to dissect these processes. So, thank you very much.